Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in this lecture, I will be explaining you absorption of dietary monosaccharides. So, in my previous lecture, I have explained you how our dietary carbohydrates are digested and ultimately converted into monosaccharides. So, here we are going to understand how exactly those monosaccharides that are coming after the digestion of dietary carbohydrates are absorbed into the enterocytes and how they are transported out of the enterocytes into different tissues and how these monosaccharides are getting into our cells and what is the effect of these monosaccharides in our cells. So, this is what we are going to see here in the absorption of carbohydrates. Let's get into the details. So, monosaccharides are almost entirely absorbed from the small intestine. That means not many of them are fermented in the colon. So, rate of absorption is 3 times higher in the proximal jejunum compared to the distal ileum. That means majority of the monosaccharides are absorbed in the jejunum itself rather than going all the way into ileum and colon. And glucose and galactose absorption compared to other monosaccharides are much faster than the absorption of fructose, mannose and other pentoses like ribose molecules. That means glucose and galactose absorption is more efficient here. And galactose is absorbed more rapidly than glucose. That you must be aware of. Okay. Now, the monosaccharide absorption in the intestine is by either sodium dependent facilitated mechanism and also it can be sodium independent facilitated diffusion mechanism. So, it is sodium dependent and sodium independent mechanism. So, 2 mechanisms are available for absorption of monosaccharide from the lumen of intestine. Okay. Now, let's first look into sodium dependent glucose transporters which are referred as GLUTs. So, sodium dependent glucose transporters, as the name says, it is all dependent on the sodium gradient that is present inside the cytoplasm. So, it's going to maintain the sodium gradient. So, sodium gradient is maintained by sodium potassium ATPase or sodium potassium pump which is present on the basal lateral side of enterocytes, which is going to use ATP. It's an active pump mechanism to pull out 3 sodiums into the extracellular matrix and pump into potassium. So, as the sodium gradient is created, so glucose and galactose absorption will be going on through SGLT2 molecule. So, SGLT2 is the one that is located over the brush border epithelium of duodenum, jejunum and ileal epithelium and thereby allowing glucose and galactose inside through sodium gradient. So, that's why SGLT transporters are referred as secondary active carrier mediated transporter because they are dependent on primary active transporter, that is sodium potassium pump. Remember that. So, that's about SGLTs or sodium dependent glucose transporters. Now, let's go into sodium independent glucose transporters, that is GLUTs. Glucose transporters, simple glucose transporters are sodium independent. That means these are bidirectional transporters. If the concentration of glucose or monosaccharides are more in the lumen of the intestine compared to the cytoplasm, so all these monosaccharides go down as the concentration gradient. Okay, and also they don't need energy, they are energy independent, they are not active transporters, they are facilitated transporters. So, glucose is transported down its concentration gradient by facilitated diffusion mechanism. So, our intestinal epithelial, brush border epithelial cells, GLUT5 is expressed which is allowing glucose to get into the enterocytes from the lumen of intestine. Whereas, GLUT2 transporters which are expressed over the basal surface of the enterocytes, it will allow glucose to exit out of the enterocyte into the portal system. So, both are glucose independent, sodium independent glucose transporter. So, from the portal blood they find their way into the systemic circulation and reach all parts of the. So, this is how by sodium independent mechanism glucose, galactose and fructose, all these molecules are getting from the lumen into the enterocyte, from the enterocyte into the portal system and carried in the blood to all parts of the body. So, let's look at what are the different types of glucose transporters that are found in different tissues. So, here in the table I have mentioned only 5 types of glucose transporters, GLUT1 to GLUT5 Otherwise, there are 12 types of glucose transporters. Now, GLUT1 transporters, as it is shown, it is present in the red blood cell, present in all kinds of barriers. It can be blood-brain barrier, blood-testis barrier, blood-placental barrier. So, all kinds of barriers express GLUT1 transporter, and GLUT1 transporters are these are high affinity transporters. Remember that. And the GLUT2 transporters usually present in the liver, kidney, beta cells of pancreas, and also serosal surface of intestinal epithelial cells. These transporters, these are low affinity transporter. That means they have high Km and high Vmax transporters. These transporters, they work only when there is high glucose levels are present there. And the GLUT3 transporters present in neurons, these are high affinity low Km transporters and making sure that brain gets glucose at all times. Coming to GLUT4 transporter. GLUT4 transporters are special compared to any other glucose transporters because GLUT4 transporter is the only insulin sensitive transporter out of all glucose transporters. Okay, and also note that GLUT4 transporters are present in the cytoplasm, and in response to insulin, whenever insulin is there in the blood and insulin binds to insulin receptor, at that time GLUT4 transporter which are there in the cytoplasm, so they will be brought back to the cell membrane and thereby allow glucose to get in. So, what type of tissues contain GLUT4 transporter? We have adipose tissue, skeletal muscle, and cardiac tissue. These are the tissues that contains GLUT4 transporters which are insulin sensitive transporter. Coming to GLUT5 transporter. GLUT5 transporters are present in the intestinal epithelial cells and spermatozoa, and it is also transporting fructose. Okay, so these are the types of glucose transporters. Coming to different types of sodium dependent glucose transporter, that is SGLTs. Now there are 12 types of SGLTs. Including co-transporter for sugars, anions, vitamins, and short chain fatty acids. So these sugar transporters include SGLT1, SGLT2, SGLT4, SGLT5, while SGLT3 functions as a glucose sensor also. Now coming to SGLT1. SGLT1 is expressed in the intestine, trachea, kidney, heart, brain, testis, and prostate. It is a prime intestinal glucose transporter. It has a poor affinity for galactose. It mediates glucose-induced incretin secretion in the intestine. Remember that. So, SGLT1, it helps in the secretion of incretin in response to dietary glucose, because incretin, it helps in release of insulin from the beta cells of pancreas also. Now, SGLT2 is expressed in the kidney, brain, liver, thyroid, and muscle and heart. And SGLT3 is expressed in human skeletal muscle and small intestine. It is a glucose sensor in the plasma membrane of cholinergic neurons, skeletal muscle, and other tissues, where it will detect a low glucose level and all that. So, SGLT2 inhibitors. SGLT2 inhibitors are located over the renal tubular cells, where it will help in the reabsorption of glucose back into the blood. So that can be inhibited by drugs called canagliflozin, dapagliflozin, and campagliflozin. These are SGLT2 inhibitors. So used with the diet and exercise, they will lower blood sugar level in type 2 diabetes mellitus, allowing glucose to enter into urine. So that's how blood glucose can be controlled. Coming to mechanism of insulin release from beta cells of pancreas. So whenever a blood glucose goes more than 3.9 millimoles per deciliter, at that time glucose gets into the beta cells of pancreas through GLUT2 transporter, and as the GLUT2 transporter allows this glucose to get in, so the glucokinase enzyme within the pancreas there, it is going to convert glucose into glucose 6-phosphate, and that undergoes glycolysis to give ATP. So, there will be rise in the ATP levels, and this ATP, it is going to go and bind to ATP sensitive potassium channel and close the potassium channel, and that will lead to depolarization of membrane. When the depolarization of membrane happens, at that time voltage gated calcium channel will open up and they will release calcium into the cell, and higher levels of calcium inside the beta cells will lead to release of insulin from secretory granules. So, along with the insulin, C-peptide will be released, and that insulin will mediate its action, and it will bring down blood glucose level to normal range. Okay, that's how is the mechanism of insulin release from beta cells of pancreas. Now, how else insulin? So this insulin, which is coming from the beta cells of pancreas, it will bind to insulin receptor, and thereby insulin signaling will go on like PI3 kinase signaling mechanism, and that will bring GLUT4s, which are there in the cytoplasm, back to the membrane. So this is how GLUT4 transporter expression can go on. Along with this insulin mediated glucose transporter expression, GLUT4 transporter expression, so even during exercise, during skeletal muscle exercise, right? So there is rise in AMP, and increase in AMP leads to increase in AMP kinase, AMP activated protein kinase, and that AMP kinase also brings GLUT4s back to the membrane, and thereby allow glucose to get inside the skeletal muscle. So this is how a glucose absorption can go on in variety of tissues with the help of glucose transporter and also SGLT1s and different types of SGLTs. So that's about absorption of carbohydrates coming from the diet and absorption of these especially the monosaccharides by peripheral tissues. Thank you.